Welcome to today's lecture. As you know, I have started a series of lectures on fish covering different aspects of fish, the spoilage, preservation and processing. So today I am going to talk about the fish spoilage in part 1. So in this I will talk mainly about the chemical and enzymatic spoilage, and there is another major cause of spoilage, that is microbial, which I will discuss in a separate lecture. So today's lecture briefly I will tell about the basis of fish composition and types of fish, and then about the spoilage aspects and details about the chemical and enzymatic spoilage. Since these lectures are specifically for postgraduate students, so I will be talking little bit more details about the chemical aspect of these topics. So briefly here we will see about the fish and its composition. As such, fish or seafoods are highly perishable due to their high moisture content and high nutritional content, or rich in all the nutrients, and the temperature in the ambient condition where it is handled, especially in the tropical countries. So if we see the composition, on an average it will have 65 to 80% water, but it varies with the species. Then there is fat about 1 to 20%. There will be fatty fish and lean fish, so that's why it is highly variable. And the protein content varies again between 14 to 20%. And there will be other important vitamins and minerals. So altogether it's a very rich source of all the nutrients, and it is highly contaminated with different kind of bacteria, and also it is exposed to atmospheric condition. Hence, it is always easily susceptible to spoilage. Here briefly I will discuss about the fatty and non-fatty fish, or sometimes called white fish. So in general there are some species of fish which has got high amount of fat, and such fishes are more susceptible to spoilage. In the diagram we can see the different aspect. If we take a cross section, in the right side for fatty fish, the side has got a portion which has more fat deposition, the dark muscle, and other part is light and it has got less fat. Similarly, in the left side, in case of white fish or non-fatty fish, it has got most of the area as light or white muscle. Only at the lateral line where there will be very little fat deposition, and in between there will be dark or red muscle. Now let us understand about the fish spoilage. We all know fish is extremely perishable, that is it undergoes spoilage very easily. And what is spoilage? So spoilage can be defined as a change in fish or fish products that renders them less acceptable, unacceptable or unsafe for human consumption. So any fish or fish products may undergo some change, and after that it become less acceptable or unacceptable due to several deteriorative changes which can cause bad odor or bad flavor or bad texture. So fish undergoing spoilage has one or more of the following signs. So there will be slime formation on the surface. There will be discoloration if we cut the section. There can be changes in texture due to proteolytic activity, and there will be off odor or off flavor again due to microbial activity and other enzyme action, and there can be gas production. So this is a brief idea about what is spoilage and what are the typical signs like slime formation, discoloration, texture becomes soft, and off odor and off flavor. So little more details about the spoilage of fish. So spoilage is the result of series of changes that happens in the dead fish, and those changes are mainly due to enzymes or due to the bacterial action, and again bacterial enzymes. So all these changes starts in fish as soon as we catch the fish and it dies. So after we catch the fish, once it die, all kind of changes takes place, mainly due to internal enzyme or due to bacterial growth and their enzyme, and sometimes due to oxidation. So in areas where temperature is high, the fish spoilage happens more early. So it can happen within 15 to 20 hours depending on the species and the method of capture. So different species, as I mentioned, have different composition, and the capture method or the water condition, all these things decide. So I am going to discuss all these separately in a lecture about the factors of spoilage. So here we will see briefly about the different factors and process of spoilage. So as such, after the fish dies, there will be biochemical changes due to breakdown of glycogen in anaerobic process and leading to rigor mortis, and it becomes stiff. And after that, the internal enzyme start acting and causing a kind of autolysis, and after that the bacteria start working, which is already present either in the gut or in the surface. So basically all the changes happens by four factors or four process. One is mechanical damage, another is enzymatic activity, then chemical oxidation, and microbial activity. Out of this, mechanical damage is related to the catching and handling of it, whereas enzymatic and chemical process continues depending on the storage and temperature, and microbial activity is a major cause of spoilage. So in this lecture, briefly I will tell about mechanical damage and full details about the enzymatic and chemical activity, whereas the microbial activity and spoilage I will discuss separately in a lecture. So in this briefly I am going to discuss about the mechanical damage and which can cause spoilage also. So this is mainly due to improper handling after catching the fish. So sometimes the fishes will be kept over the others in a large container. So the fishes at the bottom get damaged. Sometimes they may get heavy weight and damage. So sometimes there will be bruised condition on the place. So this bruising is due to the darkening by rupture of the blood vessels. There can be broken skin due to rough handling or cutting or many other things. So that can lead to the entry of the bacteria to the flesh and cause damage or spoilage. Similarly, sometimes there will be burst of guts. So due to the improper handling, there can be bursting, and that causes contamination with bacteria and enzymes to the flesh and cause damage or spoilage. So this area I am not discussing much details. It needs proper handling and training to the fish handlers or catchers. So now we will start discussing about the autolytic or enzymatic spoilage. So this is one of the most important type of or process for spoilage of fish. So first let us understand what happened to the fish after it died. As we have read in case of animal or meat animal after slaughter, so there will be anaerobic glycolysis. Whatever glycogen is present, it will undergo anaerobic glycolysis and decline of pH. And when there is no further energy available, the muscle will undergo stiffening, and that is called rigor mortis. Then later on, slowly there will be reduction in this stiffening, and slowly the pH will increase. That stage is called softening of muscle. So that is due to the action of internal enzyme. So many protein will be broken in this process, and here works many catheptic enzymes, as we have read in case of meat animals. So there will be catheptic enzymes in the lysosome that causes breakdown in the cytoskeletal protein, and there will be softening. So this is also the process of aging. So here whatever energy was there, it will be converted to ATP, and later it will undergo ADP and AMP, adenosine diphosphate and adenosine monophosphate, and finally it will be converted to hypoxanthine. So these are series of biochemical changes, and this leads to, in a normal condition, there will be better tenderness and flavor, but when it crosses a limit, it becomes a spoilage. So this hypoxanthine is an indicator of freshness in case of fish. Sometimes this enzymatic activity can cause rupture in the abdomen region, and then it is called belly bursting. Here we will see how the autolytic changes or enzymatic spoilage happens. As I mentioned, there are several enzymes in this process behind, that is catheptic enzymes, calpain, trypsin and chymotrypsin. These are the major enzymes. So what happens due to careless handling of fish, due to improper storage, no temperature control, so there will be breakdown of cells inside, and that will release the autolytic enzymes, and that starts the process of spoilage by making the substances available. So these substances create a very good environment for the favorable growth of other microorganism or for the activity of enzymes. So this is the initiation about the enzymatic process or autolytic spoilage. Later we are going to see how these enzymes work. Here we will see little more details how these enzymes work. So the enzymes I have told previously, they are mostly proteolytic enzymes. So we can call proteinase or protease. So these enzymes can digest the muscle protein in the fish or break down of the proteins, and thereby it will produce amino acids and other nitrogenous compounds. Later, by further decomposition, it will produce ammonia, carbon dioxide and other volatile compounds or basic volatile compounds like amines and fatty acids. And further action of these enzymes can produce bad smelling compounds like indole, scatol, cadaverine, etc. And all these things finally produce the foul smell of the spoilage from the fish. Let us understand the enzymatic activity which is leading to the spoilage, and we call autolytic spoilage or enzymatic spoilage. So here a number of proteolytic enzymes are found in muscle and viscera of the fish after catch. And when the fish died, these enzymes become active and cause changes, and we are telling it autolytic activity. So all these enzymes are present within the muscle or in the viscera. Due to delay in evisceration or delay in improper control of temperature, these enzymes become active and causes undesirable changes. So these enzymes contribute to post-mortem degradation, as I mentioned earlier, in the fish muscle and fish products during storage and processing. So there is a deterioration of sensory quality, and that can be contributed by proteolytic enzymes. So due to the action of these proteolytic enzymes, it produces undesirable changes, particularly in the texture and other chemical compounds, and that affect the sensory quality of fish. Now let us further understand what happens due to enzymatic activity and leading to enzymatic spoilage. Firstly, the autolytic enzymes reduces the textural quality of the muscle, and so it affects the acceptability or eating quality at the early stage. But this stage it may not produce off odor or off flavor. So this indicates that autolytic degradation can limit the shelf life and product quality, and even with relatively low levels of activity or spoilage organism. So when there is low level activity of enzymes or spoilage organism, that will affect the eating quality or texture of the muscle or meat. It may not have severe or significant odor effect or flavor effect. Now these digestive enzymes cause extensive autolysis. When these digestive enzymes cause extensive autolysis, that result in softening the muscle and rupture of the belly wall and drain out of the blood and water, and which may also contain protein and oil. So this happens when there is extensive autolysis. There will be rupture of belly and drainage of blood and other proteins and fats. So that's a more significant damage. Here we will see different enzymes which are involved in this autolytic process. So firstly, there are glycolytic enzymes. that act on the glycogen and produce lactic acid and decline the pH because there is no aerobic glycolysis, no oxygen available due to the stoppage of circulatory system. Then there is nucleotide breakdown enzymes. So these enzymes act on ATP, ADP, AMP and IMP. So first they act on ADP, then reduce to ADP, then to AMP and then inosine monophosphate, and finally it produces hypoxanthine. So that is an indicator of freshness for fish. Then there is catheptic enzymes that act on the proteins and peptides and it causes softening of tissue, but sometimes due to uncontrolled activity it may produce other bad odor compounds as I explained earlier. Then there is chymotrypsin, trypsin and carboxypeptidases. They also act on proteins and peptides and they can cause rupture of the abdominal region and it is called belly bursting. Then there is calpain, that is an enzyme that work on the myofibrillar proteins and it causes softening and tenderness, but when it causes uncontrolled then there will be more damage. There is collagenases. Those enzymes act on the connective tissue and it also causes softening of the tissue at the gap filament. Then there is TMAO demethylase. So this is a very important process. In fish there is always TMAO on the slime and due to the action of this enzyme it is changed to TMA, trimethylamine. From trimethylamine oxide it acts on the trimethylamine oxide and makes TMA, trimethylamine, and that gives the bad odor. So this is finally what happens in the enzymatic spoilage, and this is the last one. As the fish die, the enzyme activity starts and it won't stop. It continue working and leading to proteolytic changes, and that is responsible for quality loss in the fresh fish. The more these enzymes get in contact with the fish flesh, the greater the spoilage. So if there is a more delay in controlling the temperature and other aspect, the more of this enzyme will act on the fish as I have explained earlier. So it causes more damage to the quality either in texture, odor or flavor. And there is a series of chemical changes as I mentioned, finally producing hypoxanthine. So these are all about the undesirable changes due to autolytic process or enzymatic process and leading to spoilage and producing undesirable texture, odor, flavor. So far we have discussed about autolytic or enzymatic spoilage. Now we will discuss about chemical spoilage. So the chemicals present in living things are able to change due to either splitting up or joining together. So this is the background. All chemicals present in the fish muscle cells, either they may split up or join together and produce new chemicals, and they are the cause of undesirable changes either in the texture, odor or flavor, and that's what is spoilage all about. So these changes depends upon the temperature. When the temperature is high, these changes or breakdown or reactions due to enzyme are faster, so rapid spoilage happens. And another important aspect is the water availability, because water is required for all changes or chemical reactions or microbial growth. And the other factor is the enzyme. So enzymes are either within the muscle of the fish or in the gut, or it is from the microorganisms. So they are major cause for all this spoilage. So these chemical spoilage happens more in the fatty fishes. So earlier at the beginning I told there are fatty fish and less fatty or non-fatty fish. So this chemical spoilage is mainly due to the oxidation of lipid either by enzyme or by non-enzyme. So these are mostly called as lipid oxidation or sometimes auto-oxidation, and that causes the spoilage. And the fat-rich fishes are like sardine, mackerel, cutla, trout, grass carp, etc. So to further understand about the oxidative changes or chemical spoilage, so main important phenomena is lipid oxidation, and this lipid oxidation may happen either enzymatically or non-enzymatically. So when it is happen with enzyme, that I am going to see later, that is called lipolytic changes or lipolytic rancidity, and when it is purely oxidative, then it is called oxidative changes and oxidative rancidity. So this chemical spoilage process are changes taking place in the lipid fraction of the fish, and these lipids are oxidized to produce peroxides, aldehydes, ketones and sometimes other lower aliphatic acids. So the hydroperoxides which is produced through this oxidation, they do not have any taste or flavor, and it can cause brown and yellow discoloration of the fish tissue, and when the oxidative changes are very severe, that gives very bad odor and we call it rancidity. So let us understand how this oxidative deterioration or oxidative spoilage or chemical spoilage happens. So the most common chemical action which causes spoilage is the oxidative rancidity in the fatty fishes. The levels of peroxide value and free fatty acid content are both a measure for oxidative rancidity. In case of fatty spoilage, as I mentioned, due to the oxidation it produces peroxides or hydroperoxides and aldehydes and ketones. So there is a measure for that, that is peroxide value, and another is free fatty acid, which indicates the quality in terms of oxidative spoilage. So these oxidation, what happens, mostly whatever fat is there in fish, that is triglyceride, and that will be broken to free fatty acid, and from there it will produce peroxides, ketones and aldehydes. So particularly in case of fish, the lipid which is having more unsaturated fatty acid. So mostly they have got phospholipids. So these both unsaturated fatty acid and phospholipids are more susceptible to oxidative changes, and finally it produces rancid odors and color changes. So this is the background for oxidative spoilage or chemical spoilage of fish. Here we will see little more details about the oxidative spoilage or rancidity. So this is mostly pertaining to the oxidation of lipid. So it is more common or more fast in case of fatty fishes like herring and mackerel. So when there is fat oxidation, it also causes autolysis and bacterial spoilage, and high lipid concentration also contribute to the early spoilage. Most of the fat in lipid, as I mentioned, it is mainly unsaturated fatty acid, and it undergoes easy oxidation by atmospheric oxygen. And there is two factor, that is temperature and exposure to light. If the temperature is high or if there is more exposure to light, it causes increased oxidation rate and leading to bad and unpleasant odor, and finally leading to rancidity. So it will have sour or stale smell or taste. So this is the background for oxidative spoilage of fish. Here we will see further in continuation about oxidative spoilage. So lipid oxidation is a major cause of deterioration, as I mentioned, and this spoilage is more common for the pelagic fish species which are having more fat like mackerel, herring, that is having high oil or fat content. The lipid oxidation involves a three-stage mechanism and finally producing the free radicals. So this is called initiation, propagation and termination. So this is a long chain reaction producing the free radicals, and it continues, that we are going to see next. So this oxidation typically involves the reaction of oxygen with double bonds of the unsaturated fatty acid. So I mentioned the fish lipids has got more unsaturated fatty acid, and on that unsaturated fatty acid oxidation happens by the oxygen or other reasons like free radicals. Now fish lipids are not only rich in lipids, they are also having more polyunsaturated fatty acids. So these polyunsaturated fatty acids are most susceptible to oxidation. Similarly, phospholipids, which are rich in fish lipid, are also more susceptible to oxidation. So here briefly we can see the chemical process for free radical formation. So as I mentioned, there is three phase: initiation, propagation and termination. So in the unsaturated fatty acid, first hydroperoxide will react and form the free radical, then it will propagate further, and then finally it will make the free radical components, and it will continue like that. As I mentioned that lipid spoilage or oxidative spoilage happens both enzymatic and non-enzymatic. So non-enzymatic means no enzymes are involved, automatically it is oxidized. So sometimes it is also called auto-oxidation. So non-enzymatic oxidation is caused by some factors like hematine, that is the ion containing portion, like pigments, hemoglobin, myoglobin, and it catalyzes the producing of hydroperoxides. The fatty acids formed during hydrolysis of fish lipids interact with sarcoplasmic and myofibrillar protein and cause denaturation. So these fatty acids or free radicals produced during auto-oxidation of lipid can also induce the damage to the protein. Lipid oxidation can occur in fish muscle due to the highly pro-oxidative hemoglobin. So hemoglobin is a pro-oxidative agent and it can initiate the lipid oxidation, and specifically it is deoxygenated and or oxidized. It is deoxygenated and or oxidized. Finally, it is found that addition of acids which can reduce the pH can accelerate the lipid oxidation through deoxygenated hemoglobin. So if we try to add some acids and reduce the pH, it can accelerate the lipid oxidation through deoxygenated hemoglobin. So now briefly we will talk about enzymatic hydrolysis of lipid. So I mentioned earlier, without the oxidation also lipid can be spoiled due to the effect of enzyme. So those enzymes are called lipolytic enzymes, and the process is called lipolysis. So during this process, lipases, the enzyme present, can break down the glycerides. So as we know, the triglycerides, there will be a ester linkage between the glycerol and fatty acids and it forms triglyceride. So this ester linkage is broken by the lipase and we get the free fatty acid. And when this accumulation of free fatty acid is too much, then we will get the bad odor and it is called lipolytic rancidity. So these lipolytic enzymes can come from the endogenous source, either from the muscle or from the gut, or it can come from the microorganisms. So the main enzymes involved in the lipid hydrolysis are like triacyl lipase. Then there is phospholipase, which has got different variety like phospholipase A2 and phospholipase B. So this is briefly about the enzymatic hydrolysis of lipid and causing spoilage. So this is also coming under the chemical spoilage. So now we are at the end of today's lecture. Today I have explained about the basic characteristics, composition and types of fish and why fish undergo spoilage. So there are 3 or 4 main types of spoilage like chemical spoilage, autolytic or enzymatic spoilage and bacterial spoilage, of which first I have discussed about enzymatic process and how the autolysis happens and it causes the spoilage. And then I have discussed about the chemical spoilage, particularly the oxidation of lipid. It can happen with enzymes or non-enzymes, and those enzymes are either from the endogenous source from the muscle or gut or from the bacteria. But there is a most important spoilage due to bacteria or microbial spoilage, which we are going to discuss separately in a lecture. And one of the important aspect in fish spoilage is formation of trimethylamine. So fish surface always having trimethyl oxide, that is the slime which contains trimethylamine oxide, and it undergo a reduction process and forms TMA, and that forms the typical fishy odor or smell. Another important aspect in case of fish is the lipid. So many fish are high in lipid or fatty fish, and most of this lipid is highly unsaturated or polyunsaturated, and they are more susceptible to oxidation. So oxidation is a common cause, and the autolytic enzymes is a common cause of spoilage. So altogether, fish should be chilled immediately to prevent this spoilage. So which we will discuss more details about the preservation of fish. Of course, in the previous lecture, we have discussed about the cold storage of fish, either refrigeration or frozen storage. Thank you.